lecture, we'll be studying the anatomy and physiology of the functional unit of the brain, the neuron, and we couldn't find someone better for the job. With more than 5 years on YouTube, only in the past year, they launched their own website, started a podcast, and branched out into nursing videos. With currently more than 1.6 million followers from all over the world, they just keep growing. The Masters of Medical Education, and whom I'm lucky enough to call my friends. They are Rob and Kristen Beach and Zach Murphy, also known as the Ninja Nerds. Welcome, guys. What an honor.
Hey, guys. Hello.
Hi.
Thank you guys so much for having us here. We're super excited to be a part of this.
Thank you, guys.
All right, we'll go ahead and get everything kind of like loaded up here, and then we'll go ahead and get started if that's okay.
Yeah, you can share your screen now.
Okay.
On it.
So super excited, super thankful to be here with you guys all today. It's really, really awesome. Super thankful to be invited. And yeah, let's talk a little bit about the neuron. So when we talk about the neuron, obviously, it's the structural functional unit of the entire nervous system. So it's basically the foundation that we have to build on. So what we're going to do is here, we have a nice little diagram, nice little illustrations that we're going to go ahead and fill in step by step. So when we talk about a neuron, there's different components of the neuron. There's these little things, these little like tree-like extensions that are coming off of the neuron towards this region here. These little guys right there. These are called your dendrites, right? So the dendrites are kind of like the receptive region of the neuron. They're the ones that are supposed to receive signals from particular neurotransmitters. And what happens at these levels here is something very, very interesting. So if we were to take, let's imagine here that I zoomed in on like one of these little dendrites, one of these little receptive zones, and I looked at it at the cellular level, what you're going to see is something really cool. There's little neurons, like what's called presynaptic neurons, that interact with this big neuron, this postsynaptic neuron. And what happens is you release these neurotransmitters, right? And these neurotransmitters can actually bind onto these little pockets that are present on the dendrites. You see how there's like this little blue protein channel here with a little pocket? What happens is neurotransmitters will bind into that little pocket, and imagine there's kind of like a little valve or something that's blocking the channel where things can't get into the cell. Whenever these neurotransmitters bind into that little pocket, it opens up that little kind of like valve or whatever that is that's blocking the channel, and it opens it up, allowing for ions to flow into the cell. In this case, let's say that it's actually positive ions that are flowing into this cell, in this neuron, into that dendrite region. If you have positive ions rushing into the cell, it makes the cell more positive, or we can also say like less negative. And that's basically trying to depolarize the cell, make the cell more positive, excite it. And so sometimes we call that excitatory postsynaptic potentials. And that's one big thing that happens here at this dendrite, right? The other thing that we can say here that's really cool is you can also have other neurotransmitters, right, that bind onto this other channel here. Let's say that this is this green protein with another little pocket. So what happens is neurotransmitters have different functions. Let's say that we use the term glutamate. Glutamate is oftentimes a stimulatory neurotransmitter. So if it bound onto that blue protein pocket, it would cause positive ions to rush in and excite the cell. But let's say that we talk about another neurotransmitter that we utilize a lot called GABA. GABA is typically, for the most part, an inhibitory neurotransmitter. So what happens is when it's released and it binds onto that little pocket, and again, it may open up the channel, bring open that little valve, and allow for negative ions to rush into the cell. If negative ions rush into the cell, it makes the cell more negative or less positive, which is sometimes what we refer to as trying to inhibit the cell or hyperpolarize the cell. In those situations, you're trying to shut the cell down. You're trying to prevent it from having some type of action potential or graded potential. And sometimes we call these inhibitory postsynaptic potentials. These are happening constantly at that dendrite level. Now, the other thing that's really cool is neurotransmitters don't have to just bind onto these little pockets. You know what this is called? When neurotransmitters bind onto these types of channels and cause ions to either flow in or out of the cell. These are called ligand-gated ion channels. They're very, very special at the dendrite. But you can also have other channels which are indirectly activated by the ligand or the neurotransmitter. And it's another mechanism. So if you imagine at that dendrite level, you have another neurotransmitter, let's say, it binds onto this little pocket here. But this doesn't directly open up a channel. It's not a ligand gated. It's something called a G protein coupled receptor. It's crazy how so many things can happen at such the molecular level here. But you have a neurotransmitter that can bind onto this little pocket. When it does, it activates this protein inside of the cell called a G protein. And to not go down that rabbit hole, G proteins generally can activate certain types of molecules like adenylate cyclase, or it can activate like phospholipase enzymes. And basically this activates second messengers like cyclic AMP. And what happens is these things activate little enzymes inside of the cell, like protein kinases, that will go and actually phosphorylate other channels that are generally closed. But once you add a phosphate onto them, they open up and then ions will rush in. And depending upon what type of neurotransmitter, if it's a stimulatory one, you'll have positive ions rush in, excite the actual neuron. Or if it's an inhibitory neurotransmitter, you'll have negative ions rush in and inhibit the neuron. It's crazy that all of these things can happen just at that little dendrite level of a neuron. But what also is really important, not just with the dendrites as the receptive zone of the actual neuron, is a lot of this stuff can exactly the same thing happen at the cell body. So the cell body is, or the soma, sometimes we'll refer to it as, is this big honking chunk of tissue right here in the center, this big circular portion here. This is kind of like what contains a lot of your nucleus, some of the organelles like the rough ER, the Golgi, some other small things like your mitochondria, maybe some lysosomes. But there's a lot of these things in the center. And what's really cool is everything that we just talked about at the dendrite. So if there's a neuron, imagine over here acting on this guy, it can act on dendrites and try to excite or inhibit the cell. Same thing, you can have actually the same kind of proteins, these blue proteins, these green proteins, these purple proteins on the actual cell body membrane. And the same thing can happen. So I like to remember the dendrites and the cell body are the receptive part of the neuron. They receive signals from other neurotransmitters that are released from other neurons. And that can influence whether they are stimulated or inhibited. The other thing that the cell body is super crucial for is think about what it has right here in the honking center of it. It's got a nucleus. The nucleus is what basically allows for us to be able to make proteins. And proteins, when I talk about proteins within the neuron, obviously I'm talking about like neurotransmitters. I'm talking about other enzymes that are involved within the actual function of the neuron. You know, there's so many different proteins that are embedded into the membrane. Look, these proteins that are channels are G proteins. So many different proteins are involved in the function of the neuron, and it's made right here in the center of the nucleus. Right? Because you guys know basic kind of like your biology genetics is once I take DNA and I make something from it called mRNA, right? That is called a transcription type of process, right? So I'm taking a particular gene on the actual DNA and I'm kind of transcribing it and making an mRNA molecule. Once I make that mRNA, it can actually come right out of here into the cytoplasm. And what happens is it may bind onto a ribosome and then may get yanked or sucked onto this guy here, your rough ER. At the rough ER, you'll then take the mRNA, read it, and make a protein. That protein that you made from mRNA is called translation. And obviously within the rough endoplasmic reticulum, it's supposed to be able to provide some modifications to it. Maybe it'll add like a glucose molecule. It'll add other subtypes of substrates onto it, cut a little bit of it off, things like that, modify it, package it in a way, and send it to this next bad boy over here, the Golgi apparatus. And in the Golgi, it might undergo some more modifications, but be packaged eventually into these vesicles that I want to do something with. Well, imagine for a second that this vesicle, that the Golgi buds off or pops off here, it's still sitting here in the nucleus. I want those vesicles down here in the axon terminal because maybe those vesicles contain neurotransmitters. Maybe they contain proteins that I got to plug into the cell membrane. Maybe they contain specific enzymes that are needed down here for certain processes in the axon terminal. How in the world do I get that vesicle that's all the way up here in the cell body down here to where I need it to be, or in areas of the axon? How do I do that? That's a great question. I'm glad you asked. So you have this next big boy here called the axon. So the axon is this kind of like central portion here, if you will. So we have our cell body, our soma. We got these little tree-like extensions coming off of the cell body, which are called your dendrites. And if you really wanted to be particular, if some of you guys out there, you know there's a narrow portion. It's not completely shown here, but sometimes when the cell body kind of merges into the axon, there's this little small area where we call the axon hillock, and it's like the trigger zone. It's where the highest concentration of voltage-gated sodium channels are. And that's really the area where action potentials originate and start. But regardless of that, we have our axon here, this long tube which is connecting the cell body to our axon terminal, which we'll talk about last here for the neuron. What's the job of the axon? Well, obviously, one thing we know is, we already kind of alluded to this, it's going to transport things such as maybe a vesicle down to the axon terminal, or maybe my mitochondria, it's lived a good life, time's over, it's been working hard, time to retire. It's going to get sent back up to the nucleus to maybe get degraded or recycled, whatever it may be. So the axon is going to be transporting different things back and forth from the cell body down to the axon terminal. We call that anterograde transport. Or transporting things from the axon terminal up to the cell body, retrograde transport. And we use these cute little proteins here to do that. And we'll talk about what those are in just a second. But I think one of the most common things that we know about axons is that they are responsible for action potentials. They propagate electrical charges across this membrane from the cell body down to the axon terminal, right? That's pretty simple. And generally, there's two parts to it. There's a depolarization phase. And the depolarization phase, we can say, is mitigated by these purple proteins. So imagine here, in the dendrites in the cell body, I have neurotransmitters that were acting on these proteins, right? And then they got enough positive ions to come into the cell. Inside of the cell went from, let's say, like negative 70 millivolts, which is relatively negative. It's at rest at that point. And you give it a little bit of stimulus from other neurotransmitters like glutamate, and it gets the inside of the cell a little bit positive, a little bit positive, a little bit positive, positive enough that you get it to the point where this channel right here, this purple protein says, hey, baby, I'm ready to open. Let's get these sodium ions in. And these voltage-gated sodium channels reach the perfect voltage where they pop open, and then ions like sodium rush in. When sodium rushes in, it's a positive ion. If it rushes in from the extracellular space into the intracellular space, it's going to make this entire intracellular space positive. And we say when we make the cell positive, we're depolarizing. We're trying to excite it. And what happens is sodium will rush in, make this area positive, bring this to the near voltage that we need. It'll open, sodium rushes in, brings the inside positive, gets this one to the voltage it needs, and then it continues to propagate down the actual axon. Boop, boop, boop. As we move that thing down via a depolarization wave, it eventually gets down here to the axon terminal and stimulates it. And then it'll cause neurotransmitters to be released. We'll talk about that in a second. But every neuron needs to rest, right? So after we stimulate it, we need to relax it so it can be stimulated again. So it needs that time to relax. So after a positive charge moves down the axon into the axon terminal and excites this, and the axon terminal does its job, it releases neurotransmitters, etc. Then we need a negative charge to run down to relax everything. So that way, whenever it's time for it to be stimulated again, it's ready. You can't stimulate something that's already like super stimulated. So we need to relax it. And so what happens is, after this positive charge flows down, these voltage-gated potassium channels sense that the inside of the cell is super positive. And they're like, oh, too positive. We got to get this cell to chill out. So let's go ahead and open up, guys, and allow for potassium ions, positive ions, to leave the cell. So potassium will start leaving the cell. If positive ions leave the cell, you lose positive charges inside of the cell. And that makes the inside of the cell negative. And that will then cause a repolarization, because you're going to try to bring it back from positive towards the baseline resting membrane potential, like negative 70. So I'm going to repolarize it, bring it back to the original charge when it was at rest. And that's going to occur all the way down the axon as potassium leaves. Then this one will open, potassium leaves. This one will open, potassium leaves. And eventually this entire axon and axon terminal will relax. All right. So that's the big thing, is it obviously is responsible for carrying electrical potentials. Now, the other thing that the axon is really good at is this transport. And now we got to come to these cute little proteins here that are sitting here. All right. This pink protein, this one is actually a really interesting protein. It's called kinesin. And what kinesin does is it actually transports things from like your cell body down to the axon. And that would be if you're trying to transport like mitochondria down here, because you want mitochondria down here as well, right? Because mitochondria will help you to generate ATP. We need ATP to power certain types of pumps down here. So we want to bring mitochondria. So this protein will do that. The second thing is we want to transport vesicles. Remember those neurotransmitters? Here's my neurotransmitter like inside of this vesicle. I got to get it from here where it was made down here. And this protein will also transport this puppy down there. So this is a good thing to remember, is that this protein, this pink protein, will allow for what's called anterograde axonal transport, or transport things down to the axon. So that's one thing. Now this son of a gun here, this orange one, this is your dynein. And this one likes to do the opposite. It likes to transport things, particularly from the axon terminal towards the actual cell body. Now think about why that would be important. We already mentioned one of them. Maybe you're trying to take like this organelle here or other synaptic vesicles that are done and it's time for them to be over, bring them back to the actual nucleus for them to get recycled, degraded, whatever it may be. Here's another really cool thing about this one. You know, if there's like, let's say that there's some kind of injury to like the axon terminal or parts of the axon, and you lose some of the membrane proteins, right? There's a little bit of damage in the membrane and you lost some of the proteins at that portion. What can happen is there's actually something called nerve growth factor that can be manufactured down here and transported up with this protein back to the nucleus. And the nerve growth factor will tell the nucleus, hey, there's some injury down here. Make some protein so that we can repair that area of injury. And the nerve growth factor will actually get some stimulus to that actual DNA, transcribe and make mRNA. mRNA will get translated into proteins. Proteins will get packaged. I'll send them down with the kinesin and go back here to repair some of the injury. It's pretty cool, right? I think it's pretty cool. Anyway, that's one thing. The last one is where I think pathology comes into play. So oftentimes we think about viruses, not so much polio anymore, but things like rabies, herpes simplex virus, especially like varicella zoster virus. I don't know if I said it, rabies again, maybe. So even that's super rare. But viruses, they love to hitch a ride with this bad boy here, this dynein. So you know, if you get kind of like an infection from like, let's just say, for example, a varicella zoster virus. When it infects, what happens is it'll hop on and actually kind of get its way into these axon terminals. When it gets into the axon terminals, it'll then actually bind with this protein here called the dynein. And then from here, it'll get transported up to the cell body. Because why? Because viruses need our nuclear machinery to be able to replicate, right? So it'll actually get up here into the nucleus area, use our nucleus to be able to make more viruses, and then maybe it'll lay dormant for a while. And then you get older. And then for some reason, you end up with a very significant stressor in your life. You end up with immunosuppressive situation, and the virus goes from that dormant stage into an active stage. And then what happens is you start replicating, making more of the viruses, transported down the actual kinesin, and then release the virus onto the skin, and then you end up getting shingles. So it's really cool how sometimes these proteins at the molecular level can be responsible for certain pathology. So don't ever kind of like, push those off to the side sometimes. But that's the big component for the axon to think about, okay? The last component of the neuron is these bad boys, the axon terminal. Sometimes you call it the synaptic bulb, the axon terminal bulb. There's so many names for them out there. I just like the axon terminal. What happens at this region is the secretory region. So the dendrites were the receptive region. Cell body has some receptive, but primarily a synthetic function. Axon is the transport or kind of like the electrical potential area. The axon terminal is the secretory region. So we have neurotransmitters here in these vesicles that I want to get out to tell another neuron that maybe is like right here, hey, go ahead, get stimulated, or hey, get inhibited. And so how this happens is remember that positive depolarization that was running down this axon? Remember the voltage gated sodium channels? We get a little bit of positive inside the cell body of the dendrites. We open up this channel and says, hey, let's go. Voltage gated sodium channels open, positive charges run down the axon. There's this next channel here, which is like kind of like a doo-doo brown color here. This is a voltage gated calcium channel. When this gets activated because action potentials are running over the membrane, it opens up. Calcium rushes in. When calcium rushes into this axon terminal, you see these little like red proteins that look like an F. These are little snare proteins. So you have snare proteins that bind to the vesicle. So we call that your V snares. And then you have the snare proteins that bind to the membrane. That's called your T snares. If some of you guys are interested, like you've heard it's called like synaptotagmin, synaptobrevin, and then over here you got like another one called syntaxin and SNAP25. These are important in certain types of like toxins that you can actually see, like, botulism and things of that nature. But what happens is calcium gets in. When calcium gets in, it binds with these little snare proteins and causes them to link together. When they link together, kind of like a little Velcro, boom, the actual vesicle fuses with the cell membrane and then opens up into the extracellular space. and releases those neurotransmitters by exocytosis. And so that's the cool part that happens here. Now, after the neurotransmitters get released, they do the exact same thing that we talked about down here at the dendrite level. They'll bind onto either ligand-gated ion channels, and if it's a stimulatory one, it'll stimulate it. If it's an inhibitory one, it'll inhibit it. Or it'll bind onto the G protein, which will either stimulate it or inhibit it. But you guys get the point. The next thing that's really interesting that happens at the axon terminal, which often gets forgotten, is after the neurotransmitters do their job, they either need to be degraded, because we can't let them just stay there in the axon terminal continuously stimulating all day, right? We have to give our actual like target organs time to rest and then actually get a next stimulus. And so what happens is we either have to degrade them, like for example, like acetylcholinesterase, that one breaks down acetylcholine. Or another common one that we talk about a lot and we have a lot of medications for is reuptake. We bring it back into, you see this green protein, it's a reuptake protein. So after the neurotransmitter is done doing its job, it gets sucked back into the axon terminal, recycled, put back into the vesicle, and then to be used later again. You know, there's drugs, we call them SSRIs, like selective serotonin reuptake inhibitors. Things like Zoloft or there's a bunch of these different types, but all of these different types of SSRIs that we can utilize. When we utilize these drugs, what they do is they will actually work in a very interesting way to inhibit these reuptake proteins. And so imagine there's a neurotransmitter called serotonin. Serotonin is really important for our mood. And so this could be a very important neurotransmitter in certain diseases like depression, anxiety, obsessive compulsive disorder, things of that nature. But what happens is serotonin normally gets taken back up, put back into the vesicles, and they'll get released later. Sometimes in mood disorders, we want more serotonin to be present within the synapses. So if we inhibit this reuptake protein, we won't be able to bring serotonin into this axon terminal. So we'll have more serotonin stay in the actual space here to continue to keep binding onto that neuron and allow for more of that positivity and mood changes that we can see, hopefully, from those medications. So I think it's really cool sometimes to see just at like the cellular level how that can impact a lot of different diseases and a lot of processes. But that kind of covers this basic component here of like the structure and basic function of the neuron. And I think one of the cool things is now that we filled in the blanks, this is everything that we talked about. We done did it. We talked about the dendrites, we talked about the cell body, the axon terminal bulb. We went through everything here in a good amount of detail. Right. So I think it gives us the basic concept that we need to know for like just the actual basic structure and function of the neurons. But, we got to take it another step. We're not done. The other thing that we have to discuss is that sometimes neurons, we actually classify these a lot in like your anatomy, your physiology courses. We have to know the terms that may come up about neurons. So we can structurally classify neurons, which we don't do that often, to be honest with you, or we can functionally classify neurons, which is way more common of a way that we would classify or describe neurons. So when we structurally classify them, let's kind of talk about this one quickly because it's not as important. You have three ways that you can classify a neuron. You can have what's called a multipolar. All that means is if you have at least 3 or more dendrites popping off, look at this, like a bad hair day. All of these things popping off of the cell body, at least 3 or more, then you got a multipolar neuron. So if you have at least 3 or more dendrites and an actual like axon, boom, multipolar. If you have at least one dendrite, only one, and a cell body, and then you have your axon, this is a bipolar neuron. And then if you have really no like definable, this is the interesting one, there's no definable axon, there's no definable dendrite. We actually have a cell body, and then coming off one edge of the cell body, we have this portion, which is maybe coming out to the periphery. This is called your peripheral process. And then we have one going into the central nervous system. This is called the central process. But these are the 3 types of neurons. And this last one here is called a pseudo-unipolar neuron. So we got multipolar, bipolar, pseudo-unipolar. With these, the big thing to think about is multipolar, they got all these dendrites. They can pick up anything. Right? They can get you Wi-Fi from anywhere. So what happens is they can receive signals from multiple different areas. Think about why that's important. Think about your motor cortex. In order for you to be able to make a movement, your primary motor cortex, like on that precentral gyrus area, what happens is in order for that thing to be able to create a movement, you have to receive signals from a bunch of different areas. Your basal ganglia has to give you information about how to potentially allow for the exact coordination of the movement, the timing of the movement, the speed, the initiation of the movement. right? You have your other areas, your supplementary motor cortex, your premotor cortex, which are also important to give signals to that area there, the primary motor cortex. Your somatosensory cortex, which is telling you about all your sensory information. It has to tell you about the position of your muscles, your tendons, your joints in a three-dimensional space. Your cerebellum, which picks up a lot of things about coordination, posture, tone. It's got to tell this motor cortex everything. It receives all this information and says, all right, I got the perfect movement. Boom, sends it down and causes this actual movement to occur. That's all coordinated by your multipolar neurons. These are actually called your pyramidal cells, right? And then you get your Purkinje ones in your cerebellum. These are receiving information from your proprioceptive sensation area, so about telling you about your position in three-dimensional space. It receives sensation about equilibrium from your inner ears. It receives information from your motor cortex about the planned movement that you want, so your cerebellum can actually kind of like adjust it a little bit. And then from there, it can send the signal where it needs to go. So that's kind of the cool things about the multipolar neurons. The bipolar ones, I just like to remember they're the special senses, right? So your retina has them, your olfactory nerves within the roof of your nasal cavity has them, your inner ear within like your vestibule and your semicircular canals contain them. These are all for those special senses. The last one here is probably one of the more common ones that you'll actually hear a lot, especially in physiology, is you have this like a bunch of cell bodies outside of the central nervous system. We call that a ganglia, and they're right around this dorsal like horn or dorsal root portion here. And so we call this a dorsal root ganglion. That right there is this cell body, and then you have a peripheral process which may be going out to like, I don't know, let's say it's picking up information from your skin. That would be the peripheral process going out to the skin, this portion. And then it has a central process which goes into the actual spinal cord. That's the central process. And so that's one of the cool things about this one. This is actually called a dorsal root ganglion. Another one that you may see is like your trigeminal nerve. Your trigeminal nerve has a ganglion that sits out in like that middle cranial fossa. This is called your trigeminal ganglion. So if it's a pseudo-unipolar, it has to have peripheral process. We actually do. We have like your ophthalmic, your maxillary, your mandibular division there. Those are your peripheral processes. And then it comes to your cell body. So then we have to have a central process that goes into our CNS, in this part, the brainstem, where there's like this big like nucleus here. This part here going from the cell body in here would be your central process. So this is a pretty interesting way that we can classify neurons structurally. However, we more commonly in the real world classify them functionally. So you have three ways of classifying them functionally, and it's simple. They either pick up signals, whether it's a pain signal, whether it's some type of like, agitation of the skin tissue, or sensory information from your tongue, sensory information from your ears, sensory information from a muscle, a tendon, a joint, from your visceral organs, anything like that, and takes it and tells your central nervous system about that. That's the sensory neuron, or an afferent neuron, meaning it's going to the CNS. A motor neuron is going to be something that goes to your smooth muscle, your cardiac muscle, your glands, your skeletal muscles, and causes them to perform the particular function they're responsible for. But it's going out of the CNS towards that target organ. And so that's called an efferent or motor neuron. And the last one is it's in between. So it's in between the sensory and it's in between the motor neuron. It's kind of like this little guy, a little tweener. So we call that an interneuron. I think what's cool about sensory neurons a lot of the times is we can use this a lot, especially in your nervous system, is we use very specific terminology, right? So if we're picking up sensation from visceral organs, we utilize the term that's called a general visceral afferent fibers. These are afferent information coming from your visceral organs like your lungs, your cardiac tissue, your GIT, your urogenital tract. All that information is getting sent to your central nervous system via these neurons called general visceral afferent. But if it's coming from like the skin, or it's coming from like your skeletal muscles, or the things around your skeletal muscles, your joints, your ligaments, a lot of the actual capsules around that area, those things are picking up signals from that and sending it to your central nervous system. These are called your general somatic afferents. And then you have the other ones, which are basically sensory information that's coming from like your retina, sensory information that's coming from the inner ear. These will carry information to your central nervous system, but we don't like to call them general. We like to give them a special name. So these ones are actually called your special sensory afferent, coming from your eye and your ear, and that'll send it to the actual central nervous system. The last one here is if you have any sensation, for example, smell or taste, those are a little bit more visceral in comparison to sight and hearing. And so because of that, we give these a special name as well. We don't call them special sensory like these for the eye and the ear. We call these special visceral afferent information. So I guess one of the big things to remember from these is you can have afferent information that is general visceral, general somatic, special sensory, and special visceral afferent information. And that's really important when it comes down to your neurology, the basic physiology of it. With the motor component, it's the same concept. If I want to supply any motor information, so basically it doesn't have to just be movement, it could be secretory functions as well. But if I want to stimulate smooth muscle, cardiac muscle, or glands, I don't really have voluntary control over that. That's more of an involuntary type of thing. And those are usually deeper, more visceral, okay? So with that being said, it's going to be visceral efferent, but remember how this was called general visceral afferent, or in this situation, the nerves that are going to the smooth muscle around my bronchi, or the cardiac muscle of the heart, or the glands within my salivary glands, right? This information that's going to these is a visceral efferent, but is a general visceral efferent information. Same thing. If I have motor neurons that are going to my skeletal muscle, which I do have voluntary control over, these are called a general somatic efferent information. The last one, I think whoever came up with this just wanted to be funny and make, the life of students more miserable. But you have things that actually develop from the pharyngeal arches during embryology, right? So you have your first, your second, your third, your fourth. They technically get rid of the fifth, and your sixth pharyngeal arches. Those are still, a lot of those are kind of like skeletal muscle. But with the way they developed, there's particular nerves that supply those muscles of that specific arch. And so we use, like, for example, the trigeminal nerve, the facial nerve, you have your glossopharyngeal nerve, your vagus nerve. Those nerves supply very specific muscles of the pharyngeal arches. And so because of that, they got to be special. And we call these special visceral efferent fibers. And so those are usually the ones that supply our pharyngeal arches. So for example, trigeminal supplies the first one, right? Then you have your facial that supplies the second one. Then you have your glossopharyngeal that usually supplies the third one. And then you have your vagus that supplies the fourth and the sixth pharyngeal arches. So it's a pretty cool thing. So again, I like to remember general. You have your general visceral efferent, general visceral afferent, general somatic afferent, special sensory afferent, and then again, you have your special visceral afferent. For the visceral, it's the general visceral efferent, general somatic efferent, and then special visceral efferent. The last thing here is the interneurons. So interneurons, they're kind of just the little neurons that are in between the sensory and the motor component. So take, for example, like whenever you have information, like let's say that we take what's called your dorsal column medial lemniscus pathway, right? So basically it's responsible for a lot of your proprioceptive sensation, your position of your muscles, your tendons, your joints in a three-dimensional space, right? If I just kind of wanted to close my eyes and put my hands out there, I have an idea of where my hand is in a three-dimensional space. So this kind of will put everything together. So we have neurons, the sensory neurons, that will be able to tell me the position of my muscles, my tendons, my joints, my ligaments, and it'll send that into your central nervous system. So this would be kind of a what? This would be more of that general somatic afferent. And that'll go into the central nervous system. What happens is that when it gets into the central nervous system, it may synapse on little neurons in between. So it'll actually if you guys remember, this will go into the actual posterior horn, and then it'll go into the dorsal column. And then from the dorsal column of your spinal cord, it'll ascend, and get to the actual brainstem. In the brainstem, there's this one special example of a couple interneurons along the way. I just like this pathway because it's a cool thing to add on to the kind of the nitty-gritty of interneurons. As you go up the dorsal column, you stop in the medulla, and you have these like little nuclei there with little like synaptic extensions, and these are called your nucleus gracilis and your nucleus cuneatus. These are very important components of the dorsal column pathway, but they're interneurons because they pick up information from the sensory neuron, and then what they do is they relay that up to another interneuron. Okay, because we haven't got to our motor. We need our motor to be the actual component afterwards, right? So we have to have a sensory, motor. So right now I have the sensory information coming to the spinal cord, goes up here to this nucleus here called the nucleus cuneatus or nucleus gracilis. It then sends information up to another interneuron. And this interneuron is kind of a relay station for all sensory information going into the central nervous system. You know what that thing's called? It sits right here. Right in front of it is the hypothalamus. Right behind it, you have like your pineal gland. This is your thalamus, and there's tons of nuclei here. So that'll receive information, and then what it'll do is it'll send that up to your actual sensory cortex and let you know about the three-dimensional position, make you consciously aware of it, right? From there, what happens is, let's say that you want to move your arm. You want to move it in a particular pathway, right? What happens is you'll then tell your motor cortex, hey, motor cortex, I need you to go ahead and perform a movement. The motor cortex will then send information down your corticospinal tracts, come out through the spinal cord via the general somatic efferents, go to the muscle and cause it to contract. So if you think about the connection between the sensory information that was coming from the muscle, went to the spinal cord, the information that was going from the central nervous system to the muscle was the efferent fibers, but these green neurons that were in between the sensory and the motor movement was our interneurons. And so we have tons and tons and tons of interneurons. There's so many nuclei that are scattered along our central nervous system that can act as interneurons. So it's very important to remember. I think a lot of the times when we think about interneurons, though, they're commonly used in like this reflex arc. This is the classic situation. I don't know, like you're cooking something on the stove, and then all of a sudden you're not paying attention and you burn your hand. What do? You naturally like move your arm away, because what happens is you kind of provide some type of stimulus to the sensory fibers of the skin. You burned them. It causes pain, activates those sensory fibers. They send information into your posterior gray horn of the spinal cord, and then usually in between here is a little interneuron, and that little interneuron says something to the motor neurons and says, hey, the dude's cooking right now, you better go ahead and get him to move that arm off the stove. And so it activates a little efferent or motor neuron, and that general somatic efferent will send the information out and cause you to move the muscle to pull away from the actual painful stimulus. And that's really, really cool when we have these things happen. So I think that's the big thing to remember about interneurons. Now, guess what? We did all of that too. So we talked a lot about the multipolar. We talked about the bipolar. We talked about the pseudo-unipolar stuff and all the examples that we can remember from these. And then we talked primarily about the functional stuff as well. And this is the big ones to be able to remember, because this is the stuff that we commonly more utilize in the kind of like book world, and somewhat in some degree to the actual wards and in your clinical experiences as well. But that will cover our basic discussion on the structure and function of the neurons. I think one of the big things for you guys to potentially do is, if you guys want afterwards, if you want to go back in and do this, you can take this and then go ahead and try to fill it all in and just do some practice. But I think that's pretty cool. We have a lot of stuff on our actual website if you guys want to check that out, about a lot of other topics besides the classic neuron anatomy, structure, and function. But we have a lot of other notes, illustrations, and videos that cover many, many other topics as well. Again, I just want to say I'm super, super thankful to be able to be here with you all today, to be able to talk with you guys. It was really, really enjoyable. And if you guys have any questions or any concerns or anything at all, I think now would be the time to be able to talk a little bit.
Thank you, Zach. Yeah, we got plenty of questions. So
Okay.
we'll go through the ones that we can for the next like 15 minutes or so.
Okay.
So first question, it says, unrelated, but as first-year med student, what attitudes and skills should I develop in order to not only do well in school, but to become a good doctor?
That's a great question. So I think one of the things that I try to live by in the clinical practice, I work in a neuroscience ICU, is to stay humble. Don't you're going to be learning a lot of information. So I say, first off, stay humble, and then don't get discouraged. But I'll explain why. So when I was in school, I remember just having to learn massive amounts of information in a very short time period. And the same kind of goes for all of you guys as well. And that can be very stressful, because you feel like when you get out there in the clinical world and you're taking care of patients, you're like, oh man, I don't feel like I know a dang thing. I don't feel like I remember anything that I learned in school. And that can be super stressful and sometimes discouraging. But I would say just be positive, keep working hard, keep trying to study and do the best that you can at all levels. So as you guys go through your first year, just do the best that you can and focus more on not memorizing tons and tons of information. I know this is probably something that's repeated to you like multiple times, but do your best to understand the information, truly have a good basis for it. If you can find out the reasons why, I think that's always the good question, is why is this happening? Why is this the way that it is? I think that's a good kind of like skill tool to always kind of have behind you whenever you study something. And again, I think that applies to the clinical world. Whenever you guys are out there and you feel like you don't remember something, you don't know something, don't get discouraged. Just ask yourself why, and then go looking for the answer. Put in the time, put in the effort, and always put your patients first. I think the other thing is, maybe you feel like you know a lot. Maybe you feel like you're really prepared and you remember everything that you learned in medical school. That's great, but stay humble. There's going to be some situations that you may have a lot of information in that big cerebral cortex of yours, and all those neurons are packed with information, but sometimes you may get thrown for a loop. So be humble and always be willing to admit when you don't know something, search for the answer, and always be willing to take in like advice from other people. Don't be prideful, because I guarantee you there will be something where you will get messed up and forget something, and the patient will actually pay the price for that. So always put your patient first.
Thank you. That's a great advice. I think we can now use it even if we're not in first year anymore. So another question, it says, how is the sensory somatic nervous system involved in sensing information and motor function?
So I'm sorry, what was the question again? Sorry.
How is the sensory somatic nervous system involved in sensing information and motor function?
Modern function. Oh, yeah, I think it's involved in everything. Obviously, with our somatic nervous system, all of the sensations that we have that we experience on a daily basis. So obviously, I'm sitting on a chair right now. I'm completely aware that I'm sitting on a chair. And that could be because of a lot of the proprioceptors that are present within my muscles, the tendons, the joints within my body. And so that's a conscious awareness that we have that are involved within our somatic nervous system. Pain. So obviously, if you think about the somatic nervous system, there's a lot of different things that we can actually feel. Crude touch, pressure, pain, temperature. A lot of different proprioceptive sensation, fine discriminative touch. I mean, if we could go on and on and on about how all of these things are involved in our modern day lives, but I think that's relatively obvious when it comes to, again, how these things are involved in everyday life. Everything that we do, whether we're sitting, whether we're talking, whether we're moving, everything comes into play with those sensations. I don't know if that was a good enough answer.
Yeah, it was perfect. Thank you. Here's another one. How does myelination affect the propagation of an action potential?
Oh, that's a great question. Yeah, so when we talk about myelination, you obviously have these different cells that are, a part of your glial cells, your glial cells, however you'd like to pronounce them. I still don't know which one's right, but either way, you have the oligodendrocytes and the Schwann cells. Obviously, those are super important with myelinating the axons of neurons. Obviously, your oligodendrocytes more for the CNS and Schwann cells for the PNS. But with that myelination of those axons, they basically provide kind of an insulatory function and allow for concentrated voltage-gated sodium channels to be between those areas of the actual myelination. So if you have like a myelinated area, that will occur there, and you have a little space between the next myelinated area. And those are usually called your nodes of Ranvier. And usually in those nodes, you have a super, super high concentration of voltage-gated sodium channels. So whenever the electrical activity moves within that area of myelination, it moves super fast to the next concentrated area of voltage-gated sodium channels. They open, sodium rushes in through those, and again, travels super fast through the insulated area that's covered by the myelin. So obviously, myelin is really important for being able to speed up action potentials. And the more myelinated the neuron is, the faster the action potentials is. And that's basically through that type of conduction. It kind of looks like it's skipping along the neuron that you call it saltatory conduction. So yeah, myelination is super important. Obviously, why in certain diseases such as, Guillain-Barré syndrome or multiple sclerosis, there's certain alterations in those functions because of the decreased kind of like movement of those signals because of the demyelinated process that's occurring.
Thank you, Zach. Yeah. So here's another one. In what way does an increase in neurotransmitters, like quantity, affect the amount of information passed from one cell to the next?
So yeah, that's a great question. So again, when we talk about like like pository, like excitatory neurotransmitters. Classically, we think about like glutamate. So I think one of the best examples of something like this is when it comes down to like seizures. Generally, in certain situations, when you have like maybe like this excitatory activity, there's lots of glutamate that's firing for some reason. So if your neurons are releasing tons and tons and tons of glutamate, that's causing the neuron that the glutamate is actually binding onto, those receptors on it, to allow for a lot of sodium and calcium ion influx into the cells. And if that's occurring, you're getting a lot of positive ions rushing into the cell. They're depolarizing, they're firing, they're generating a lot of action potentials. And if that's occurring, and it's occurring in a very rhythmic and consistent pattern, you can obviously see people having seizures. And so sometimes what we can do is we can give them particular medications, right, that target those glutamate receptors. So things like ketamine. Ketamine is a common one that we can utilize that can act to be able to kind of work against those receptors where glutamate binds to be able to kind of decrease the excitability of those neurons and potentially help to break seizures. And the same component, you can have people who have the exact opposite. Like, gamma aminobutyric acid is also important. So whenever that one works, it basically helps to be able to allow for particular negative ions to be able to rush into the cell, to hyperpolarize the cell, to arrest the cell. Well, maybe in certain diseases where they're having issues where what happens is you're not having those GABA actual ions being released. The GABA molecules either aren't binding onto the receptors properly, but that pathway is dysfunctional in some way. And so what can happen is sometimes we can utilize particular drugs. For example, in seizures, if you want to break a seizure, you can increase the GABA activity within the synapses or increase the GABA receptors. And so we can give drugs like benzodiazepines or propofol and things of that nature, because what it'll do is it'll help to increase either the GABA concentration or increase the activity of the GABA receptors response to GABA, and we'll get more negative ions rushing into the cell, inhibiting the cell, causing the cell to basically hyperpolarize or relax, which will allow for the action potentials to decrease and help to be able to drop some of that seizure activity. So I don't know if that kind of helps with the answer, but yeah, sometimes too much excitability is a problem. And I think sometimes we can target that, especially in these diseases like seizures.
Thank you. We have another one that I really like, actually. Okay. So it says, what is a mirror neuron?
A mirror neuron. Is that what you said?
Yep.
Oh, I have no idea. I wish I knew what a mirror neuron was.
All right.
Cool, but I don't know.
Okay, maybe it has another name in English. I'm pretty sure. And it's like the literal translation from Spanish.
Let me look it up real quick in the interim. If you want to find another question, I can look it up.
Yeah. Okay. We can go to the next one and then go back.
Okay.
So what type of neuron is the most common?
Oh, yeah, that's a good question. So I think when it comes down to it, a lot of the times with these, your multipolar neurons are super, super common. Obviously, you have like a lot of those within your central nervous system, but you also have tons of interneurons. I think it's kind of a combination between those. But I think with multipolar neurons, obviously a lot of our activity is dependent upon being able to receive multiple signals. So if you have multiple dendrites to be able to pick up multiple signals from multiple areas of multiple functions of the body. So again, we use that example of the primary motor cortex. You have to have neurons like those that respond to multiple signals of different sensory information as well as kind of pre-programmed motor information. And so I think that's really important to have lots and lots and lots of those. But there's also tons and tons of interneurons that are scattered along our kind of like central nervous system as well. So I think, yeah, those are kind of the big ones that I would say are going to be the most concentrated types of neurons that we would see within our central nervous system.
Okay, and last one. Do brain cells reproduce in the healthy adult?
Sorry, you said, what is it?
Do brain cells reproduce in the healthy adult?
So generally, neurons, like unlike other cells, are generally like amitotic. So usually, whenever neurons are injured, they don't generally replicate and make new cells. You may repair certain neurons, but you don't generally like completely remake an entire neuron. That's why whenever patients develop like massive strokes or bleeds within the brain, a lot of the times what actually happens is not necessarily that their neurons regenerate or they actually like make new neurons or they replicate new neurons. neurons, oftentimes it's actually something called like neuroplasticity, where they develop kind of I guess the best way of saying it is like a compensatory mechanism. Other neurons that are actually healthy will kind of take over or help in assisting to provide some type of regain of function to maybe if you stroked out a large territory of the right MCA territory, now you're actually having some left-sided weakness. Some other neurons may actually kind of take over or compensate to help regain some of the strength back in that left side. But it's not necessarily that you actually replicate new neurons, make new neurons, and all of a sudden your left-sided weakness completely resolves. So I would say that generally the most common kind of like basic biological concept with these is that generally neurons are amitotic. They can repair, but you can't completely like replicate another neuron.
Yeah, right. Thank you. We got one more question. Can we go through it?
Yeah, let's do it.
Yeah. So it says, how does herpes zoster affect the neuron and how is it related to facial paralysis?
Yeah. So whenever you talk about like these types of like viruses, whether it be the varicella zoster or whether it's like a herpes simplex virus or something of that nature, generally what happens is you get some type of infection where it infects the nerve terminal, the axon nerve terminal. And when it gets into that axon nerve terminal, it'll hop on to the actual, again, those proteins, the dyneins. And then it'll actually travel up the actual dynein proteins into the actual cell body area, where it'll actually utilize the nuclear machinery to be able to, again, make more viruses, and destroy parts of the actual facial nerve. And so that's why you can develop facial nerve palsy, and there are certain situations there because of certain types of viruses that actually, again, hop onto the axon terminal, use those motor proteins like the dyneins to transport themselves up to the cell body, use their nuclear machinery to replicate, make more viruses, and then obviously via that like lytic cycle, they can cause destruction of the actual facial nerve, particularly multiple neurons of the facial nerve. And then obviously as you lose function or lose some of the nerves that are components of that big facial nerve bundle, you'll start to experience weakness of that actual facial muscles.
Awesome. Thank you.
Yeah.
So that was the last question, and we want to thank you very much for your contribution. Seriously, we are very thankful, and we feel greatly honored to have you guys. And yeah, that's pretty much all.
Well, first off, it was super awesome to be able to be here. Super thankful, super blessed. Again, you guys could have picked a lot of other people besides me. And so the opportunity to be here, to talk with you guys, to provide kind of a basic concept or conceptual topic on the neuron, I think is a blessing. And so I'm very thankful to be here. I hope I was able to answer some of you guys' questions, and I hope you guys had fun. And just thanks for allowing me to be here. And I hope you all have an awesome and amazing day.
Thank you. Thank you very much.
All right, take care.
Muchas gracias a todos por habernos acompañado en esta primera ponencia.